All right, so just a reminder, in our photosynthesis chapter, we've kind of split this up into 4 parts. Our first lecture really focused on the chloroplast and looking at a lot of homology between the chloroplast and the mitochondrion. Now we're going to move on to the first part of photosynthesis, which are the light reactions. And then our next lecture will move on to what are referred to as the dark reaction or the light independent reactions. And then our last lecture will focus on regulation. So again, just a reminder that this light reaction really uses light energy to excite electrons. And so we're going to see a lot of homology and a way to review electron transport in mitochondrion. So we're going to see 2 reaction center chlorophylls or photosystem complexes here called 2 and 1 that allow us to use light energy to promote electrons to a higher energy state. We're going to see homologs to coenzyme Q in plastoquinone, and then we're going to have a homolog to cytochrome C here in plastocyanin. And so we're going to see something analogous to the Q cycle with cytochrome B6F, which has homology to complex 3 in mitochondrion. And where things get a little bit different here is where we have this ferredoxin protein and then this ferredoxin NADP plus reductase enzyme. So again, this is the final destination of electrons. But this is, again, where something is a little bit different here in that we form NADPH as the final electron acceptor is reduced. And what's kind of going to be interesting that we'll talk about a little bit towards the end of this lecture is there's the opportunity once electrons get to ferredoxin to either go this way, or they can actually go back to the Q cycle and go through this pathway again, pumping more protons and generating an even greater proton gradient. And so kind of like we saw with the pentose phosphate pathway, depending on what the cell needs are, you could either kind of siphon things to go towards ribose synthesis, or you could have it go towards NADPH synthesis. So in some ways this is similar, that if we just kind of go straight through, we're going to pump a proton gradient, but not an excessive proton gradient, and in that case the focus would be NADPH production. The cyclic pathway will allow us to pump a greater proton gradient, making more ATP, but then again at the sacrifice of making NADPH, because the only way you make that NADPH is by the final delivery of electrons. All right, so kind of a lot was said there. We're going to have then this last part, which is not called oxidative phosphorylation, it's called photophosphorylation, and that's because we use that proton gradient generated from the kind of photoinduced electron transport chain to generate ATP. All right, so that was a lot kind of on that first slide here. But again, the 2 purposes of the electron transport in chloroplasts are to make NADPH by reducing NADP plus as those electrons get to their final destination, and then also generate that proton gradient, which we can use to make ATP. So in our activity, and big thing that I want you to be thinking about is homology between what we've seen in electron transport in chloroplasts and then reviewing what we've seen in electron transport in mitochondria. So again, we talked about this in our activity in our previous lecture, but just to kind of reinforce it here, when we think about the origin of electrons, the idea in mitochondrial electron transport is those electrons come in a high energy state just because of their chemical nature being part of NADH or FADH2. In chloroplast electron transport, the low energy electrons are what we start with in these chloroplasts or the chlorophyll pigment molecules. They become high energy using photons of light. And so we generate those high energy electrons. And we can kind of see this in what they call a Z scheme. And the reason they call it a Z scheme is it kind of makes the letter Z if you kind of turn it on its side. But we have a very similar plot to what we saw here when we showed NADH all the way up at the top, giving electrons all the way down at the bottom to oxygen. So we're looking at a standard reduction potentials. And remember, in order to go through electron transport, we have to have things that are very likely to give up their electrons, giving their electrons to molecules that are more likely to take those electrons, and we keep kind of stepping down. That happened chemically in mitochondrial electron transport just by virtue of the nature of those molecules. What happens with chloroplast electron transport is we have these electrons that get excited to this high energy state. So you can see here this electron in P680. So that's the wavelength of light that that chlorophyll molecule absorbs. That's why we call it P680. So when that chlorophyll pigment molecule absorbs light at 680 nanometers, it excites an electron up to an excited state. So that's what we have when we have, say, P680 star. And in that excited state, what that really means is we've changed the standard reduction potential to be very negative. So that chlorophyll really wants to give up its electrons. And again, it'll pass those electrons through a series of carriers that allow us to get it to our next carrier, which is again plastoquinone here. Taking it over to cytochrome B6F is what we have here. And again, going through the Q cycle will allow us to pump the proton gradient. But remember here, and part of what we'll see here on our next few slides, is as that electron leaves this excited state chlorophyll, it needs to be replaced. So ultimately the electrons for chloroplast electron transport do come from water. So we have the splitting of water into oxygen, the oxidation of water to generate molecular oxygen, and then those electrons. Those electrons again are going to be replaced, that electron lost in that chlorophyll molecule, and it happens in the oxygen evolving center. So ultimately we do have electrons that come from water, but they originate in that chlorophyll pigment molecule. Again, plastocyanin, analogous to cytochrome C, is going to take those electrons from cytochrome B6F over to complex photosystem 1 complex. And now we have a chlorophyll that has a different wavelength of light. We call this P700, again excited by a photon of light, going through a series of carriers. And we'll spend time talking about this cyclic pathway. But once these electrons get to ferredoxin, there's the opportunity to continue to the FNR complex to be reduced, to have NADP plus be reduced to NADPH, or those electrons can return to plastoquinone or to that Q pool and continue through the Q cycle again, pumping an even greater proton gradient. So what we're going to have on the next several slides is we're just going to kind of highlight little pieces that we have here. And then we'll again show it in comparison to this figure here. So we can kind of map on where we are on each of these figures. Not going to spend a lot of time going through this in the overview. We'll spend the details, we'll cover those details in our activities. But again, electrons start in photosystem 2 with that P680 chlorophyll. They're excited by light, and then they pass through a series of carriers onto our plastoquinone. We again have 4 protons that are going to be pumped or introduced to the thylakoid lumen. That's that intermembrane compartment. Again, moving them out of the stroma. And again, they ultimately end up, the electrons end up at cytochrome, or at this plastoquinone, our Q analog here. So we ask the question, how does P680 plus, that cationic chlorophyll molecule, regain its electron? And that happens from the splitting of water at this oxygen evolving center. So there's the oxygen evolving center, again, the oxidation of water, generating molecular oxygen and releasing those electrons back to that chlorophyll. I'm not going to spend too much time talking about the oxygen evolving center, so don't worry about this S0 through S4. We're not going to cover the details of the oxygen evolving center, but again, that's how we replace those electrons that were lost from the chlorophyll molecule. Moving those electrons from the Q pool onto the cytochrome B6F complex. Don't really need to spend too much time covering this either, because this is very analogous to what we saw with complex 3 in mitochondrion, and again we learned the Q cycle. So very, very homologous in mechanism for translocating protons, again to the intermembrane compartment. Not too much necessarily to say here. We'll talk about some of the carriers that we have there in our activity, but again, this transport very similar to what we see in complex 3 in mitochondrial electron transport. So at the end of complex 3, we delivered electrons to cytochrome C in mitochondria. Now we're going to deliver those electrons to a 1-electron carrier called plastocyanin, or PC. So again, we're going to have those electrons get delivered then to photosystem 1 complex. Once we're in photosystem 1, those electrons are again low energy, so in order to have them go through a series of carriers, they're once again excited by a photon of light. This time it has to be a wavelength 700 nanometers in order to match the absorption pigment, or the absorption necessary for that pigment. Again, we're going to go through a series of carriers, but this becomes unique once we get to ferredoxin, is there's the ability to have that ferredoxin continue on to FNR, or in a cyclic pathway, have the electrons return to the Q pool. So again, we're going to have ferredoxin or the Q pool, very similar here, if they go back to the Q pool, to what we see here, repeating the Q cycle. Again, that's not an option in mitochondria. We don't have the ability to go back and ride the ride again. So again, two possible fates for electrons. Non-cyclic pathway just has those electrons continue from ferredoxin over to FNR and generate NADPH. The cyclic pathway is going to have those electrons be redelivered to a pool of plastoquinone molecules, and those will then be allowed to go back through the Q cycle and generate an even stronger proton gradient. So again, the needs depend on whether you're favoring NADPH production for the non-cyclic pathway or ATP production for the cyclic pathway. So assuming you've continued at some point along the non-cyclic pathway, electrons will be delivered from ferredoxin to this FNR, this ferredoxin NADP plus reductase enzyme. Those electrons are delivered to their final electron carrier in NADP plus and generating NADPH. One slide on this for our ATP synthase. It's not called an F1 F0 ATPase, it's called a CF1 CF0 ATPase because of the chloroplast origin. But other than that distinction, this machinery operates exactly the same as we saw in mitochondria. So identical structure and function to what we saw in mitochondria. So we really don't need to spend too much time going through the details. So a couple of things, and again, our activity will allow us to highlight and recall these. A few things are very unique to chloroplast electron transport compared to mitochondrial, but one of them is the ability of the membrane to be permeable, that inner membrane that we have in chloroplasts, analogous to the inner mitochondrial membrane in mitochondria, that is permeable to ions like magnesium and chloride. And what that allows is for electrical neutrality. So remember, when we pumped protons in the mitochondrion, that generated an electrochemical gradient. We had not only the difference in concentration with H+, so that created a pH gradient, but we also had a positive charge on that inner membrane space side of our inner mitochondrial membrane. Wow, there's a lot to say in this chapter. So the idea is we have to have a greater pH gradient in order to have the same thermodynamic driving force. Remember, the magic number was about 3/4 of a pH unit in mitochondria. It's going to be 3.5 pH units in chloroplasts. And again, don't worry about any of these numbers, but again, final tallies allow us to have a certain number of protons translocated per oxygen generated. And so we're going to, in some ways, think about having a PO ratio again. So recapping what we have here, right, we've got electrons coming in ultimately from water, but beginning with the chlorophyll molecule, passing through photosystem 1 to plastoquinone, participates in the Q cycle in cytochrome B6F. Plastocyanin takes those electrons to photosystem 1. Additional light is going to excite those to allow them to pass through a series of carriers, ultimately residing then in ferredoxin. Depending on whether we're having a non-cyclic or a cyclic pathway will allow us to focus on the production of NADPH, or in the cyclic pathway, generate more ATP through ATP synthase. So that is the light reactions. So remember, the purpose of the light reactions are to use light energy to excite electrons to go through electron transport and generate a proton gradient that's used to make ATP. And then the final electrons that we have there, I got to go all the way back to the end here. The final electrons that we have there are going to be used to make NADPH. So that's the net outcome of the light reactions. As we move into our next lecture on the dark reactions, we're going to see that those two species are used to help do what's called carbon fixation. So that's taking carbon dioxide and using those electrons essentially to glue and reduce those carbon atoms so that we can make sugars. So we'll move on to that in our next lecture, and then our final activity will be how do we regulate all of this, particularly when we have to make sure that whether it's light out or dark out, we're doing different things with plants.